Hey comrades, today I welcome you to my YouTube account. We are doing discussion about medical virology. This is to help any medical student who is having hard time in classifying viruses, how to approach them. I'm here to help you out. Virology is the study of viruses, and majorly viruses are those living organisms that have got this ability of living in both a living state and non-living state. They can survive as living organisms or non-living organisms. And viruses are in living organisms, like when they are inside a living organism, other living organism like an animal, a plant, a human being, they exist as real living organisms. But now when they are exposed to the external environment, the harsh external environment, they undergo into non-living state. And in that state, they always appear as prions or particles. Classification of viruses is based generally on their genome or like genetic makeup. And viruses can be classified as DNA viruses or RNA viruses. Unlike for us humans or human beings who have majorly DNA as our genetic makeup, the viruses for them can either live as RNA or DNA. That is to say that you can find a virus having DNA as its genetic makeup or RNA as its genetic makeup. Back to our classification. Today we discuss major RNA virus classification, so that next time we can handle DNA virus classification. RNA viruses are either single-stranded or double-stranded. And we have to note that whether single-stranded or double-stranded, their genetic material is either always encased in a nuclear envelope, or it is always naked, just lying naked in the cytoplasm. So there are those viruses which have their genetic material enclosed in the nuclear membrane, or like others, they have just DNA material just lying freely in the cytoplasm. Now, our RNA viruses can be classified as single-stranded or double-stranded. The single-stranded RNA viruses, they are either positive sense or negative sense. Positive sense viruses, for them, they have this ability when they are replicating. They directly undergo replication, produce daughter virus cells. But for the negative sense viruses, they don't have that direct replication ability. They will always need an enzyme called RNA dependent RNA polymerase for conversion into positive sense. I am trying to say that the negative sense, for them to be converted to positive sense, the genetic material of these viruses, for it to be converted from negative sense to positive sense, there is an enzyme required for that stage. And that enzyme is called RNA dependent RNA polymerase enzyme. We continue with our classification. Remember we started with RNA viruses with single-stranded or double-stranded. When you go the single-stranded RNA viruses, we have those which are positive sense or negative sense. The positive sense, we are trying to mean RNA. RNA is either positive sense or RNA is negative sense. For the positive sense RNA, it can either be enclosed in a nuclear membrane, that is genetic material genome. It can be enveloped or it can be naked. The viruses with enveloped DNA material, most of times during replication, they replicate through a DNA intermediate. And there are those which have the ability to replicate through the DNA intermediate, and those which don't have that ability. And the viruses with the ability to replicate through DNA intermediate, they include the genus of Retroviridae. And those which don't have the ability, we include the genus of Togaviridae, Flaviviridae, and Coronaviridae. Back to the positive sense virus, we have the virus that lack the ability. We have the virus that have naked genome, or like the genetic material is not enclosed in the nuclear membrane, and they are positive sense also, and they include the piconaviridae, the calciviridae, astroviridae, and hepaviridae. To continue with our classification, in the single-stranded, we also have the negative sense. The negative sense for them, they will include Floviridae, Bunyaviridae, Rhabdoviridae, Osomixoviridae, and Arenaviridae. We should note that, as I had said before, a negative sense virus, for it and complete replication, it must have to be first converted into a positive sense virus, and that process requires an enzyme called RNA dependent RNA polymerase enzyme. That's why I minded much putting it for you at the bottom of this classification, so that you could always have it at the back of your mind. In the final phases of classification, we have discovered that RNA is either, the RNA of viruses can either be classified as DNA viruses or RNA viruses. And for RNA virus classification, we have discovered that the RNA can be either single-stranded or double-stranded. And then, for the single-stranded RNA, we have discovered that it can either be positive sense or negative sense. That's how we come up with deep and deeper classification of various viruses types. Now, for the double-stranded RNA viruses, we have those with naked RNA and those with enveloped RNA. And for this, we are only lucky that we have only the viruses which are RNA double-stranded and have naked genome material, and that includes the Reoviridae. I hope this classification will help you out and will simplify your work as you try to enjoy the medical course. Thank you so much.